clusters, I mean, I think it's not really applicable then. And also what you can see is that, for example, cluster 1, you have this color here, and I think it also wouldn't really make sense to have a seed zone which is so far apart from each other. So I still need to figure out to find a good way how to delineate it into different seed zones. Yeah, and here you can see the genetic offset calculations. So for future climate, we use the climate data averaged for 2041, 2070. This is the average obtained from 13 global climate models for 3 different shared socioeconomic pathways, which are also currently used by the IPCC, so that's why we use this data set. And you can see the darker the color is, the higher is the genomic offset. And we use different scenarios, like from the best one to the worst, again, on the example of the BioInf data set. And you can see, yeah, that with a worst scenario, we have a higher genomic offset. And what I find also surprising me that we have this really dark spots in Alaska. So, but this method still has some limitation. I mean, we only take the environment and the genetic information, and which is not going into the model is like the type of reproduction. Like we have clonal propagation in Mexico, and it's not accounting for this. As well as the distribution shift in species competition influence or hybridization with other species. Then it's also not accounting for like phenotypic plasticity, epigenetic changes, or polyploidy, because we have more triploids in drier region what we observed. So my further steps are to complement the model maybe with some phenotype-associated genes. So for a subset of the individuals, I have data from a greenhouse experiment, so differentially expressed genes under drought. Not sure how to incorporate it, but I think it's a good idea to put this into account. And because there are no existing ecozones for Populus tremuloides, maybe I look for co-occurring species and look which ecozones are already there. And I think it's also a good idea to use some additional programs to confirm if similar clusters can be found. So yeah, if somebody has a nice idea maybe, I'm happy to discuss it. And with this, I would like to end my talk and thank my co-supervisors and all the funding agencies, also my contact details if you want to contact me. And because I am the last one to talk, I would like to take the opportunity to thank the organizers for this nice conference. For me, it was really nice, and yeah, thank you really much for making this happen.
Okay, so I think we have time for 1 or 2 questions, if there are any for Melanie.
Thank you very much. I was wondering, did you explore the possibility of building a model just with the environmental data? Without using the genetic data at all, and then seeing if that was any different, like just, you know, kind of like a null?
Yeah, I mean, it's possible to just plot the environmental data to see just the differences in like temperature, precipitation. I mean, that's possible to make this also visual, like the genetic composition. Yeah.
Thanks for your presentation. I think I remember from the presentation that you chose 900 non-clonal individuals. I was just wondering how you determined that they're non-clonal and why non-clonal?
Yeah, non-clonal so that we don't have the same genotype twice. So when we had the same genotype twice, we excluded one. Yeah. I hope it answers your question. Okay, great. If there are no further questions, I
think it's time we wrap up this session. Let's thank all our speakers once more.
Okay. Well, first I'd like to say a big thank you to our keynote speakers and our plenary speakers for putting together some wonderful talks for us today, or this week. Let's give them a big round of applause. And also a big thank you to all the students and postdocs and researchers that gave talks this week or posters. We really appreciate it. It made this conference a huge success. So let's thank them as well. Thanks. And I think there's one thing that we learned from this conference that you're going to take with you for the rest of your life, and that is don't be the last one in the lunch line, because you might not get anything to eat. All right, since I'm currently the president of the WFGA, I'd like to say just a few words really quickly about that organization. This concludes my obligation as president, and I'm now going to relinquish that role on to Dr. Jill Hamilton, who's now going to take over. Jill was at North Dakota. She was in North Dakota when she became the vice president and agreed to become the next president. But since then, she's moved to Penn State. And if you know anything about U.S. geography, Pennsylvania is not in the western United States. But she said she's determined to help us organize the next conference. The next conference for the WFGA will be in 2025. And we're looking at combining with SIFTIC, the Southern Forest Tree Improvement Cooperative. So stay tuned for more information. We're trying to breathe a little bit of life into this organization. Unfortunately, over the last few years, we've had declining participation in conferences and meetings. For whatever reason, we're going to try and breathe a little bit more life, so we're trying to include more people on the executive. So if you're a young postdoc or student or young, good-looking researcher that just gave us a wonderful talk from UBC who might want to be involved, Tom, let me know. We're looking for people to help get involved. We might try and start things like a speaker series to bridge the gap between conferences. So if you're interested, let me know. It's a great group. It's very informal, nothing too formal or anything like that. We have a good time. We get together and we talk about forest genetics, which is awesome. Okay, just another few acknowledgements before I pass it on to Andreas. I know we're all waiting to hear about the results for the student awards. Andreas is getting everything organized for us. I just want to say another big thank you to the field tour organizing committee, in particular Marie Vance, who was on the committee, helped out quite a bit, and she's still helping us one more for the big field tour tomorrow, as well as Krista Copeland and Dan Gadet. They weren't here this week, but they also helped quite a bit. And Trevor Dirksen, who is going to lead us tomorrow, and if we get lost, it's his fault. Another big thanks again to Dan Gadet and the Vernon Seed Orchard Company for the banquet, and Andreas for doing the most important job of organizing the judges and scoring the students for their awards. And on that note, I'll pass it on to Andreas.
It's definitely not the most important job. I just added the numbers, that's all. So, thank you for letting me present this. We should thank the judges first because, I've served in this role before, and you have a very different conference experience if you're appointed as a judge because you have to pay attention, you have to think about everything. You learn a lot more at those conferences. And this time we had 20 student presentations and 8 posters. I think in terms of presentations, this might be close to a record, This is a lot. That's great to see. Now we broke this up a little bit to make it not too onerous. So we have quite a few judges, and I'll just read them out. Ashley Thompson, Andy Benowitz, Andy Bauer, Dave Calotello, Deogarcias Rayongeza. I should be able to say his name because he's a very close collaborator of mine. Keith Jojo Kruma, Jason Holiday, Patrick Lenz, Sally Aiken, and Tom Booker. So let's thank the judges. We do have 2 first prizes for posters and 2 first prizes for presentations, because they are sponsored respectively by the Canadian and by the Western Forest Genetic Organization. So I start with the CFGA. And I feel like I should say something about the sponsors of this. So we have the Jean Namkoong Award for Best Poster, $500. Jean Namkoong was a very, very influential forest scientist. He got appointed in the 1970s as a pioneer researcher with the USDA, a very rare appointment, many honors in his career. And he, the thing that impressed me most, he came actually to UBC in the 1990s, and I attended his retirement party. And that was probably one of the biggest parties I've seen from, he really touched the lives of many people across the world. There was almost from every continent, there was somebody there. That was amazing. So that award goes to M.I. Hayward, University of Victoria. Please stand up and, yeah. You actually only need to stand up. We got nothing for you, so there's no cash at the moment, but we will get in touch with you. Very good. Karl Heimburger Award, CFGA Award for Best Presentation, goes to, oh, yeah, I should also say something about him. He's considered the father of tree improvement in Canada. He got started with the first provenance progeny trials in the 1940s, also with the Canadian Forest Service. This prize is 1,000 Canadian, goes to Adam Gilewski from Simon Fraser University. And then we have the WFGA awards. They are in the name of William B. Critchfield. He was equivalent pioneer in the US. He got forest genetics started in the 1960s with the US Forest Service in California. Also very influential. Lots of tests that are out there bear his association or name. And we have a Best Poster Award from this category that went to Rayani Diaz, a familiar name from U of A. Rayani? And the Best Presentation Award, US $700, goes to Sam Grubinger, UBC. There he is. Very good. That's it. Thank you very much.
Thank you, Andreas, and congratulations, students. We do have one more award, and Raju from the Canadian Poplar and Willow Council of Canada is going to present that award. And then after that, I would like all the students to come up on stage and we'll get a picture with you together, okay? Raju?
Thank you, Brian. Hello, good afternoon, everyone. I'm the current chair of the Poplar and Willow Council of Canada. And at these forestry meetings, we always encourage our graduate students to come and deliver talk on poplars and willows and other fast-growing trees. In the midst of conifers, we need to show up ourselves. So generally, we give annual travel bursary award for graduate students. This year's winner is Aini Diaz Saliva. Please come forward, Aini. Yeah. Yes. I have a certificate for you, and check will be a $500 given to you later. Thank you all.
So would the other students please come up here, and we'll get a picture of you together. The award winners. Come on. Sam, Emma, Adam. Okay, right. Yeah, can we get them in front of the screen? If you just maybe just drop the brightness of the screen down a bit. Okay, we'll get a white backdrop for you. Okay. Thanks. Can you pull this? Okay. I get to wrap it up. Okay. I would also like to thank our sponsors for their generous support financially and in kind. You wouldn't believe the costs associated with these meetings. So it really helps. Otherwise, you would have been paying thousands of dollars to be here. Well, you probably did when you added up your hotel bill anyway. So thank you very much for taking your time and your own money or your employer's money to come all the way here to join us, because this interaction in person is really valuable, especially for the students. If you're only on a 2 to 3 year program, you need these opportunities to present your work and get familiar, see others, and aspire to be up here one day as a keynote speaker yourself. So that's great. I'm glad the students had an opportunity to be here. And for the CFGA to be able to support the undergraduates that are just embarking on their careers and inspiring them to continue on. So thanks for joining us. Yes, the acknowledgement slides next as well. I want to thank particularly the people that helped us with, organizing the meeting behind the scenes, in particular UBC's conference and accommodation services, and Sonia in particular. You've been corresponding with her. She's been emailing me over the last couple days asking how it's going, and I've flipped her a few pictures. So if you go onto the home page, you'll actually see pictures of our banquet dinner, a few other things. If you see yourself and you don't like it, we can take it down. But we'll post a few more pictures up there from the conference. And as I say, we were recording these sessions. Thanks very much to New Horizons Productions. So I would like to give a big round of applause to Ben and Bryn and company for their support. Really makes a big difference when you got some professional technicians and some nice equipment as opposed to us running around with our little portable projectors and figuring out how to load presentations at the last minute on a laptop that's not compatible with someone's USB drive. And you've all know that feeling, right? And then the black screen of death. Yeah, that's great. And I'd also like to, they're not here, but I wanted to maybe we clap loud enough to thank the hotel and their staff for hosting us the last few days, and for the meals and the snacks, etc. So a big thanks to Prestige Hotels. Yeah, thanks Don and Brendan, and of course the front desk staff. Yeah, we had great service from our, well, just about all our caterers, and then, yeah, I think that covers everyone, right? Is that good? Yeah, just gotta pat ourselves. Okay, we'll get out of here. Okay, yeah. So we're gonna record we have recorded the session, so we'll send you a notice of when they're posted online, so you can catch up on the other concurrent session you didn't attend. People that had posters, they're lying on the floor in the Okanagan room. So if you want to roll them up and take them home, otherwise they're going to go into the recycling bin. And I think that is it. For those that are on the tour tomorrow, it's 8 8:15 in the lobby. 8:15 in the lobby, and then we'll get sorted for vehicles. And right? Anything else on that? We're good? Okay, 8:15. Okay. All right. Well, so I do have a couple more slides and some jokes, but can I go one more? One is just to acknowledge my colleagues on the board of directors of the Canadian Forest Genetics Association. Their names are here, a few in the audience. So it's been the last couple of years, this is, we got signed up in 2019 in Quebec, and we ended up doing a 4-year stint instead of 2. But what is different now is in the last couple of years, we have registered our association as a nonprofit organization with the federal government. Despite its origins going back to about 1936-37, it has not been like a legal entity, organization. So now we have legal status, so we can pay taxes or GST, and also we can apply for insurance and enter into contracts, so that make it a legitimate organization. We're also shifting away from just doing an informal business meeting to actually now we're obligated to have an annual general meeting and maintain membership lists. So the AGM of the Forest Genetics Association will happen in October. It'll be the 2nd annual, and so it'll be the members there. One of the agenda items is to update our bylaws so we can clearly define what our membership is. In the past, it was you needed to be nominated by 2 people at a business meeting. Of course, at business meetings, it was typically only those people that were able to travel to that meeting, that biannual meeting. So we're going to update that so there will be opportunity for anyone that's interested in forest genetics in Canada to be able to become a member. So we'll give you more information later, and we would welcome people as guests to the AGM. But of course, the members are the ones that get to cast the votes. So that'll be in October. And then I can, like Nick, I can then maybe pass the baton to someone a little further east from here. And so that hasn't been determined yet as to where the CFGA is going to host its next meeting. I will put it out here publicly that it looks like it's Ontario's turn, but we'll see if they've got, and otherwise we'll look at where we can go next. So until then, thank you very much, and I wish you safe travels home, and it's been a pleasure. Thanks very much.